Testing, one, two, three. Okay, good. Welcome to this session of Shadow the Scientist. My name is Jamaica Marshall, and I'm very pleased to serve as the Shadow the Scientist coordinator. Shadow the Scientist is an initiative housed under the Creating Equity in STEAM umbrella of programs offered by a team at the University of California at Santa Cruz. We offer free online public experiential learning sessions with professionals in a variety of STEAM fields several times a month. On the screen, you can scan our QR code to explore our website. There, you will find a link to our YouTube channel, register for upcoming sessions, request a dedicated session for your specific group, or even request to host a session to share your science. So check us out. In this session, we have the immense privilege of shadowing the members of the Lico Na Polina team, Rebecca Ostertag, Emma Stierhoff, and Sophia Montoya. Loosely translated, Lico Na Polina means building or growing new partnerships or relationships in the Hawaiian language. This project name reflects their goal to restore degraded Hawaiian lowland wet forests by creating hybrid communities using both native and non-invasive, non-native species. I'd like to welcome Rebecca Ostertag, the principal investigator of the Lico Na Polina Hybrid Ecosystem Projects, to get us started. Welcome, Rebecca.
Good morning. Welcome. We're really glad you can join us today. We're going to show you about our lab today and some of the things that we do. We are an ecosystem ecology lab. So let's talk a little bit more about what are the actual things we do in the lab. Broadly, we study tropical forest ecosystems. And so we want to understand how forests are structured and also how they function. So for structure, we look at things out in the forest, like what species are there, what are the size of the trees, what are the abundance of the different species. And that structure is really important because you've got big trees or small trees, dense things, open areas, high light, low light. That influences all the kinds of different species that live there and the species interactions, the food availability. So the structure of ecosystem, we say that begets their function. They're intimately related, okay? But we also do a lot. Most of what we do is a lot about how the forest functions. And this is what a lot of ecosystem ecologists do, is we want to understand the health of the system. And we think in a very systematic, holistic way. So we're like doctors of the forest, but we're thinking about the whole organism, all the organisms, the forest as one big kind of superorganism, and how the different pieces fit together, how they do and they do not work together, and different kinds of interactions. So one of the main things that we look at in this lab is nutrients and how nutrients move around from soils to the plants and then back to the soils. Because we're very interested in that. Nutrients are the food of what the plants are taking up from the soil, and then all the animals and the detritivores are eating that food and releasing it and recycling it. And that, I mean, what's more important than food, right? That's the most important thing to life. So that's a key thing we study. We also sometimes study how water moves in the forest. And we also look at how light influences plant growth and forest structure. So those three things, nutrients, water, and light, are what needed by every single plant on Earth, okay? Everything that it photosynthesizes. So that's even more than just plants. But so we critically need to understand how those resources, call those abiotic resources, because they're not the living components, they're the non-living components of the ecosystem. We critically need to understand how organisms use them, how they compete for them, how it relates to their health. And so we study, this is kind of like the basis of what we study. And then we need to know what a healthy system is, but we also study a lot about the perturbations. So one of the key things in Hawaii, we have a lot of species that come from elsewhere. More of our flora now is from somewhere else outside of Hawaii, i.e. non-native, than within Hawaii, native. And some of those species are highly invasive and are having major effects on growth and reproduction and the health of the forest through nutrient cycling and water use and light availability, and some aren't. And we want to know how different perturbations affect the forest function and structure. So we look at this and we know that species are all different in how they, even though they all need the same three things, light, water, and nutrients, they vary in how they use it. So every species has their own sort of formula and range in which they can use these different abiotic resources, metabolize them, and use them for growth, reproduction, and defense. So that's the kind of stuff we want to understand. And then once we better understand how the forest functions, how it works, especially by studying these native and non-native species, then we want to figure out, after these perturbations, how can we restore the forest? How can we, after we have a lot of invasion by non-native plant species that are highly invasive and changing the environment, how can we get it back to something or move it to a different state that might be in a healthier state? So we have to understand all these different aspects of health. There's no normal anymore. In what we're studying in today's world, humans have been influencing the environment in so many different ways. So, but we understand these different modalities of health. And forest, one forest may be healthy in one place, and that same species mix may not be healthy elsewhere. So we really have to do this on a case-by-case basis. So how do we do this? So in the field, we're going out and we're measuring the health of the forest. So just like you go to the doctor, we're doing a checkup on the forest. So Emma and Sophia here go out in the field quite a lot. And once a year, we, in our Liko Napolina experiment, for example, we take data on what species are there. We have plots, we have the trees that are permanently tagged and marked, so we know who each individual is. And we go out and we measure their diameter. That gives us an estimate of their size, okay? And so how big are they? How much have they grown since their last checkup, which was the previous year's survey? So we can calculate that based on the information that we take out in the field. And then we look at what are the species that are there? Are there new species that have come in? What's died? How has that mix sort of changed? And then we do the lab tests, the health indicators, just like when you go to the doctor for your physical, they take blood and with that blood, they're going to maybe do a cholesterol check. They're going to check your sugar levels, see if you're diabetic or pre-diabetic. Maybe they're checking your cholesterol. All of those things, we're doing that with the plants. And sometimes we do that with the soils as well. But just different indicators. But we use this lab work as indicators. So that's the kind of stuff that we do in the lab. Some of the health indicators that we look at are litter fall. So Sophia's going to go through today what we do with the litter fall. The litter fall is all the dead leaves that have fallen on the ground in a given time period. So we measure that out in the field. We have trays that we put out in the field, collect that litter fall. Every time a plant makes a new leaf, it drops an old leaf. So that litter fall is basically an index of how productive that forest is, how much is it growing. And we want to know that. We want to do that. So what we do is we collect all that litter, and we'll show you the ovens in a minute. We dry it and we weigh it to get an estimate. So we don't have to count every single leaf, but we get a weight. That's our sort of estimate of the amount. And then we do this grinding in fine powder of the leaf samples, and then we send them to a lab for nutrient analysis. And that gives us the percent nitrogen and the percent carbon. These are the two most critical elements for plants, for plant life. Nitrogen is used in amino acids, proteins, enzymes. It's a primary nutrient that's needed for photosynthesis in the enzyme rubisco. It's also in chlorophyll. So plants cannot grow without nitrogen. They also can't grow without phosphorus. Phosphorus is used in ATP, adenosine triphosphate, which is the fuel source for all metabolic processes. It's also used in the phospholipid membranes around cells. So if you've taken general biology, this might sort of ring a bell. If you don't have fuel, you can't grow, reproduce, defend yourself either. So those are two really key nutrients. That's checking on the health status. This is Emma's going to show you more about the data analysis. Once we have all this data, how do we know how healthy the species are, how healthy the forest is, what's going on, where are they in their cycles? We study this all year round. So sometimes during the year, more things are going on than other times. We look at all of that the whole forest as a whole system. How is it doing and how healthy is it? I'm going to show you now another thing that we do, and this is more at the species level, is that we want to understand species, how each species uses these resources, light, water, and nutrients. And that's what we call a functional trait analysis. So functional traits are things related to how a species uses water and nutrients. So right here, for example, I took some plant samples from outside. Here's one. Here's another. Okay. And here's the third. And you can see like this one, super, super thick leaves. This is going to affect how well it photosynthesizes. Thicker leaves are not going to photosynthesize as well. Look at its color. That color relates to the amount of chlorophyll it has. We look at things like the leaf size. We're going to measure leaf area. So this is a really big leaf. This is a smaller leaf. These are even smaller leaves. How much area a leaf has. We look at the leaf stem, the petiole, and this has to do with the display of the leaf. So these are like floppy leaves that are going to be open to the sun. These leaves have no petiole and they're more upright. So which one's going to get more direct sun beaming on it? Okay, this one's going to be collecting the sun more than this one. Okay, also that petiole and how the leaf arrangement is going to relate to how much water they lose. How conservative are the plants in using water? So we do that as well. We look at those kinds of things. And I'm going to show you a couple slides as a background. Yeah. Okay, so once we figure out the functional traits of the species, we put it in a functional trait analysis to look at how species occupy trait space, principal components analysis. And I'm just showing this graph as a simple way to get it. But each species is different. So these are fruits, okay? And there's two axes. Tasty to untasty is the y-axis, and the x-axis is easy to eat and open and get the good parts. And difficult on the other end. So pineapples are really tasty, but they're really hard to like get to the pulp and get to the good juicy parts. A peach is very tasty. It's not so hard to open that up and get to the pulp. Okay. And so these fruits all fit in different places in what we call trait space. And that's what we're also doing with our plants. We want to see where they are. We're going to measure, we measure more than these two things. We measure about 15 different traits. We want to see where each species falls to get a sense of how they use the different resources. So here's just an example with 15 traits that we measured in the past. So each dot is a different species. The blue, native species are in blue, highly invasive species are in red, and non-native species that are not invasive are in this gold color. And you can see they kind of separate. The native species kind of are in one part of the graph. The non-natives are in another part of the graph, and the invasives are kind of everywhere. And we can see also on the axes where they separate. So those arrows there are showing what are some of the key traits. So foliar nitrogen, and the leaf mass per area, which is getting at the leaf, how much leaf area there is per mass. So it kind of gets at leaf thickness. It's a key trait. That's separating on the x-axis. And on the y-axis, things like the mass of the seeds, how tall the trees are in the field, and how much phosphorus they have in their leaves are separating on the y-axis. So we do this kind of stuff to get a sense of where all these species fit. And then when we do restoration or want to build communities, we want a balanced community. So we don't want all the species to be kind of in one corner. We want to get a balanced species where they're going to be working together. They can use the resources available in different ways. And then hopefully that allows for better coexistence and overall forest health. So I'm going to show you some examples of what we do and the kind of data that we collect. Is we would go and we have a master spreadsheet here of all the different things that we measure. And we would start looking at the leaves. And from the leaves, we would get information. So one of the first things we do is we would just separate our leaves, cut them off here, and we start doing some measure. Okay, that's a trait that we're interested in, and the petiole length. And we were interested in this ratio. How long is this leaf stem relative to the length of the leaf? So this one has a pretty small petiole relative to the leaf. This one here has no petiole. Okay, and then this one would have a very long. This is actually a compound leaf. And so this is got 3 leaflets on it. And this would be a very long petiole. So this is going to be a floppier kind of leaf display. So we would do those kind of measurements. We would also take this tool that's called a micrometer. And we would open it up here. And we would put in our leaf. And this will measure the leaf thickness. So it's a digital micrometer. There. This is a very, then I tighten it just enough, and then I'll get a reading of its thickness in centimeters or millimeters. I think this is millimeters. Yeah, millimeters. Okay. And so this leaf is obviously going to be a lot thicker than another leaf. So this kind of leaf, I would choose, and I would want to choose a central part and not on the main mid vein. So, and this one is so tiny that I would have to be really careful about where I chose to measure it. But it's obviously going to be way, oh, see, it's not even cooperating, but it's obviously going to be way thinner than the first leaf. And so I would get it in there, measure the leaf thickness, take that data down, and then I would have that. Then the next thing I want to do is I want to know the area of these different leaves. So I would take my whole sample, and I would, after I made all my leaf measurements, I would collect each leaf separately. I would cut the petiole off at this point because we don't want it anymore. And then I would measure the leaf area. And so this machine is a leaf area meter and should stabilize at some point. So we calibrate. It's got a special calibration tool. It's got a calibration tool of a known area, and we run this through ahead of time to calibrate it. This is 10 centimeters squared, so we would run that, and then reset here, and we would put our leaves in here. This is basically just like a fancy camera instrument underneath, but I add up my leaves, and it's giving me the leaf area. So for my sample here, I would then write down that I had 3 leaves, and this would be the leaf area. So then I would have that information, okay, with the leaf area meter. And that's really crucial because that's giving me information of the size of the leaf, how much photosynthesizes. Then I would want to get my leaf dried and weighed. So I would label it, and I would put it in a paper bag. And I would put the proper labels on it, and then I would stick it in our drying oven over here, just set it 70 degrees, oven, and you would put it in here, and I would leave it in here for at least 48 hours. And the reason that we do this is to get the actual dry weight. And the dry weight is something that's comparable around the world. So a leaf here in Hawaii is going to be, it's a lot more humid here than, let's say, a leaf in Arizona or in the Atacama Desert in Chile or something like that. So we want to know globally, we all do the same standard, get the water out of the leaf. And then once the water gets out of the leaf, then we weigh it again. So that's going to take at least 48 hours. Then I'll weigh it, and that will be the dry weight. And then I'll do some calculations, calculate how much area there is relative to the dry weight. That's leaf mass per area. And that's an important calculation. So we do a bunch of different things on the leaves. Then I'd also do some things on the stem. So here is a stem from this species here that I cut off. And I want to measure its density, wood density, or so-called stem specific gravity. So we soak the stem for about a half an hour. So I've had it soaking in here. And in order to measure the specific gravity, we actually do a volume figure out the how much, there's a certain weight of water in here. And when I stick the stem in here, how much is this water is this stem going to displace? And I can weigh that in grams on the scale. And when I do that, then I can do calculations to get how much volume was displaced. And so I would put this on the scale. I would tare my scale, and then I would add my stem. And now the tricky part is that I want my stem to be like completely underwater, but not touching the sides. Okay, so it's kind of like a balancing act to get this to work. You can use 1 or 2 of these things, but I don't want it to touch the sides because that's going to influence the weight. It's got to be free floating. It's kind of got to be sunk down in there. So, oh, see, this is always a challenge. This is the high tech part, but you get the idea. We want this to be free floating, not touching the bottom or the sides. Okay. And I just need to have the pin go down at it. Close enough. Okay, you get the idea. It's going to be free floating in there, and then we'll take the weight, and from that, we'll be able to calculate the density, the specific gravity. And from the specific gravity, what we get is like trees with denser wood grow slower. So this is something about their growth, also how they're using water and nutrients. So we know that they grow slower. Get there's seed size databases. We collect things on seed size. We collect things from the literature on maximum height of the plants. So all of these things go into our giant spreadsheet to do that principal components analysis. And then with that information, we know more about the plants and we know how each species is different and what kind of resources they tend to use and they tend to favor, what kind of conditions they would do the best in. So we use that information when we're designing restoration strategies to help pick which species are going to do the best. We want species to do best in a given environment. We know the environmental conditions more or less, but we also want to know species that will play well together. So we don't want species that are going to be too, one might completely outcompete the other. We want to have a more balanced community or a more balanced team. So that's what we do. So that's the functional trait component. And I'm going to turn it over to Sophia, right? She's going to talk about litter fall.
So after we get all the leaf litter out of the ovens, because they are set at 70 degrees Celsius, not Fahrenheit, they're always coming out nice and warm. But before we even put them in the oven, we have 20 of these trays out in every plot. So we have a total of 20 plots, which is 4 repetitions of each combination. And then we collect them, dry them at the oven, and then they come back. So part of our job after we get back our dried leaf samples is we get a bulk weigh. So we take the whole sample of the bag and weigh it. After we get that bulk weigh and we notate it down, we have to sort them by species. So what we do is we go off the plot. So for instance, for this demonstration today, we have plot 9. So it's going to be a moderate redundant plot. And then these are the species that we are going to sort for. So sort of partly already sorted, we have our ulu, false kamani, avocado, mango, this is our scioto, monkey pod, and then this little guy is our little rose. So we go out and then we have to identify and sort the whole sample. We do get some that are not part of the out plants. And this is what we discard. So like this is a copico leaf. So he just doesn't get counted for now. And then once we get done with all of the sorting, we go all the way down to our little tiny boys. And some of them, they get a little bit difficult to sort. And so you have to look at like defining characteristics. So like this is a scioto leaf and it has like this rounded shape, but at the same time, it's really shiny. So mostly all of their leaves will have this really nice shine to them. So there's like little tricks that you'll find to help you sort and differentiate a lot of the species. But once they're all sorted, we go back to our scale and we'll weigh each species. And after each species is weighed, we'll label our bags, add them into our bags, and then they get sent off to be grounded. So when we go to grind our leaf litter, it gets grinded really, really fine. So I'm not going to turn the machine on because it is very loud. But we insert our leaves to the top, there is a spinning blade in the center that will spin and cut it, and then we have our filter. So in our filter, it has a mesh top, and this mesh top will only allow our grinded leaf litter of a certain size. It's like this one is set at 40. And then what it's going to give us is this really fine leaf litter, like the leaf litter groundings. We package them up all nice neatly with what month it is, the plot number, and then our identification number. And they get sent off to the lab on campus, which then will light them on fire and then get the nutrients that we're looking for out of them. And so that's our nutrients from that leaf litter. What we get from the lab. So after we collect things, she has how we put everything together from that.
Oh, there we go. Okay, thank you for bearing with our technical difficulties. Aloha kako. My name is Emma Stierhoff. I am the outreach coordinator / technician for Liko Napilina. And so I'm going to be showing you today some of our data analysis side of things. So Becky and Sophia told you part of how we collect our data, and they focused more on leaf litter. What I'm going to be focusing more on is phenology. So phenology are these natural cycles that living beings go under. And so we focus on the fruiting and flowering of our trees. So every month we go out in the fields and we collect data on worksheets that look just like this. You can see that. So we take these worksheets out and basically we have a subset of trees in each of our plots. And we look at if those trees are fruiting or flowering. And so we just put yes or no for fruiting or flowering. And then we compile all of those yes and no's into a much larger data set. And so I will show you a bit of what we can do with that. Share my screen here. Okay. So this is our master data file. It has phenology from the start of this project in 2014 up until this goes until 2022. I'm going to show you analysis from the year of 2021. So you see every month we have this data collection. We have our trees tag number. Let me go up to the top to show you that. We have who collected the data. We have the treatment for the plot. So these treatments are either slow or moderate, and that's based on the analysis that Becky showed you, as well as the analysis we get from the lab with those nutrients after leaf litter grinding. And so that tells us if a tree is cycling nutrients slowly, so maybe it's not dropping quite as many leaves, or maybe it has much smaller leaves, so it's not putting as much nutrients into the system. Whereas moderate nutrient cycling would be a tree with much bigger leaves, or that's dropping more leaf litter, and so it's cycling more nutrients, or maybe it has more nutrient dense leaves. So we're looking at all of those things, and so we're determining if a treatment is slow or moderate based on the combination of plants that we put there. And then we also have redundant and complementary. So redundant means that you have more trees with similar functional traits. So after all those analyses that Becky did, you can determine what types of functional or roles those plants are playing in the environment. And then you can determine, are they playing more similar roles or are they playing more complementary roles? And so redundant would be that you have more plants with similar functional traits. So if you think of it like a soccer team, you have more plants playing defense than you have playing offense. You have more plants with redundant roles. Whereas complementary is you have more plants playing different roles. So it's a bit more balanced in terms of the roles they play. So complementary would be a soccer team with balanced offense, defense, midfield, and a goalie. And so part of this project is looking at how these plots fare based on those treatments. So one thing that we are prioritizing, like I said, is phenology, because that tells us if plants are reproducing or not. A plant can't reproduce if it's not dropping any fruits or any seeds. So we look every month at if a plant is fruiting or flowering, and that tells us if they are actively reproducing or not. And so that all gets compiled into a master data sheet just like this. And then once we have that master data sheet, we can start doing some analysis on it. And so I tend to do my coding in R. That's what I'm most comfortable with. But there's a number of different coding softwares you can use. And so I'll kind of walk you through how I compiled this data. I'm just going to focus on the year of 2011. There are ways you can do comparisons across years and all of that, but that gets very complicated very fast. So we'll try to keep it simple for today. So I have these two packages that I uploaded into R that we're going to use. It's dplyr and lubridate. Don't worry too much about what that means. So the first thing I'm going to do is import. Actually, let me clear everything out from earlier. So I'm going to import my data first. So that's this read.csv. So that's taking that file I just showed you and putting it into R. And then what I'm going to do is narrow down this whole giant subset. You can see there's 37 something thousand variables right now, or rows of data. So we're just going to focus on 2021. And so this next line is taking that subset of data and making it just 2021. So now we're down to 4,722 rows. Then I'm going to add a row for the date. So if you take a look at this file, we have date as a month, date, year format right now. I want to get just the month because we take data once a month. And so I want to be able to show how our phenology changes from month to month. So that's what this next line does using that lubridate package. So next, I am going to add a column that summarizes how many species are flowering and fruiting. So right now, our data is set up that we just have each individual species. So we do have the species name, and then we have the tag number and what plot it's in. But I want to know how many of these are yeses within each plot. So I want to know how many of each species says yes for flowering and how many says yes for fruiting. So we're trying to summarize all these Y's and N's into something meaningful. So that's what this next line is doing. It's grouping by plot and by month. So for each plot, so say for plot 1, which is or actually we start with plot 2, because plot 1 is a control, and that's a slow redundant treatment. I'm asking it for this plot and for the month of January, how many species are fruiting? So it's telling me of all of our species, how many of those are fruiting? And I'm having it do this for every single month and every single plot. So that's what that group by function does. So we run that. And then I also want to get the proportion of flowering species, because some plots just have more species richness than others, or more total species. And so it can be more meaningful to look at the proportion of species that are flowering than just those raw totals, because it's unbalanced in that way. So I'm getting another column with that proportion with this next line of code. Then I'm doing the same thing, but with fruiting this time. So I'm getting how many yeses for each month and each plot by species are fruiting. And then I'm getting the proportion as well. So now I have this phenosum data set that I just created. And this has additional rows, or additional columns, sorry. So this is showing how many species are flowering within that plot and within that treatment. And then it's showing the proportion and then how many species are fruiting and the proportion. But now I have a bunch of repeats for each plot and each species. So I want to make it so that for each plot and species, I just have one. Or sorry, for each plot and month, not each plot and species. So that's what this is doing. It's selecting the unique month and the unique plot, and it's giving us just one row. And so you can see, let me show you more here. This pheno unique has far fewer rows than the pheno sum. So it's making one unique row for each of those observations. And then I'm just doing some kind of data management stuff so our future code works. So you have to kind of tell R what to read your columns as. So right now it's just looking at our month as a number. And so I'm telling it to look at it as a month rather than just a random number. So then we're going to use this ggplot package to create some graphs. And so I'm just going to create a box plot. So we have 4 duplicate blocks, each with 5 treatments. So we have a slow complementary, slow redundant, moderate complementary, and moderate redundant, and then a control for each block. And then we have 4 blocks as replicates. And so we have 4 replicates of each treatment. And so I was curious, looking at those 4 replicates, how does phenology change over the course of the year? And then specifically looking at species richness. So I want on my x-axis the month of the year, and then on the y-axis, I want how many species are fruiting or flowering. So that is what this is doing. Hopefully, there we go. So this is a very, very busy graph because this is giving me for every single month, each treatment, how many flowering species there are. And it's showing all those 4 replicates. So these box plots are showing the maximum flowering species, the minimum, and the median. So it's giving me a lot of information and it could be useful, but it's also really hard to interpret. Sometimes more is not better with graphing. If you put this in a paper, people would be probably very confused. It is useful information as you're interpreting your data and trying to figure out what's going on with our plots. We can kind of start to see some trends in phenology over the year, but it's a little too busy for my liking. So I'm going to try to simplify it a little bit. And so this is specifically looking at flowering here. So I'm going to make a new document that takes the mean of those 4 replicates for each treatment. And so rather than having that spread of data, we'll just have one mean value to graph. So I'm going to do that and then create a new plot using those means. So now we have this much cleaner looking graph, and it shows you by each treatment how many species are flowering for each month. And so now we can start to kind of discern some trends. For example, with the slow complementary shown in that blue, it's flowering a little bit. There's some species flowering at the beginning of the year, but then as the year goes on, in the months of June and July, you start to get quite a lot of flowering species. And then it drops off a little bit, and then you get another little surge in October. And so you can do that with each of these treatments now. So for slow redundant, you have very little flowering species at the beginning of the year, and then it starts to go up, peaks in May, then it goes down again, then it goes back up at the end of the year. So in October, November, December, we're starting to get more flowering species in those slow redundant plots. And so it's interesting just to look at these different trends in flowering as the year goes on for these different treatments. But as I mentioned, we are not balanced in our total species in each of these plots. So I want to look at the proportion of flowering species, not just the totals. And so that's what this next line of code is going to do, is give me a graph showing those proportions. So now we have a graph showing the percent of species that are flowering throughout the year. And we're seeing similar trends, but it's giving us a more balanced view of our plots. So next, I want to do the same for fruiting species. So flowering is telling us if they're maybe getting ready to reproduce, but they can't reproduce unless those flowers get pollinated and then produces these fruits. And so fruiting is also really important to us. So this graph is going to show the total number of species that are fruiting over the course of the year. Again, we can start to discern some unique trends across the plots. For example, looking at the moderate complementary plots, it's fruiting quite a lot at the beginning of the year in January. Then it starts to go down, and then it comes back up around July, goes back down again, and then we have a peak around October, November, December, and into January again. So it seems like in these moderate complementary plots, we're getting the most number of species fruiting kind of towards in the winter time, like October, November, December, January. Alternatively, looking at the moderate redundant plots, which is shown in that red, we have much lower fruiting species at the beginning of the year, and then it comes up around July. So that peaks in fruiting a bit earlier in the year than it would for the moderate complementary plots. So these are just some trends that we can start to discern from making these graphs. And then once again, I'm curious to look at the proportion if that differs. So I'm going to make one more graph showing the percent of species fruiting by month. And we are kind of getting some different trends popping up here. You still have the moderate redundant a bit lower at the beginning of the year, and then it pops back up in July. But this gives us kind of a better idea of a balanced sample and how many plants are fruiting. So that is a big crash course on our data analysis, but hopefully this gives you an idea of what we can look for, how we can turn this data into meaningful information, and how we can interpret that information as well. So we have a few minutes left, so I'd be happy to take some questions for everyone, if anyone has questions that have popped up during this session.
First, I'd like to say thank you all for taking us through your lab today, showing us exactly what you do here after you come back from the field. It was absolutely fascinating to see all the different components that go into your research. I'd like to start with a question about your R programming. I've never seen R in use before and only recently heard about R. I know that it's mainly used for traditional statistical analysis, but do you ever use Python? Python, with an astronomy background, Python is something that I'm, of course, much more familiar with than R. And I've never having used R, I'm curious to know if there are any similarities between the two and if you ever use Python.
I've actually never used Python personally. I'm kind of a newbie when it comes to statistical analysis, at least relatively. I've used R quite a bit at this point. I used it quite a bit in my master's thesis. So I feel very comfortable with R at this point, but I haven't explored too many other coding languages outside of R.
So I got a question that was sent to me earlier from a guest who had to leave and go to another meeting. So the question was about how you engage with the community, and in particular, getting your research out to middle high school students and university students who may be interested in interning with your team in order to contribute to this kind of community research, because it's so vital for our ecosystems.
Yeah, so Sophia and I have been doing a lot of work on that in the last several months, and we're kind of picking up steam with our outreach efforts. So in the previous year, and we're going to continue this collaboration this year, we've worked with folks from the Ka'umeke Ka'eo Charter School, which is a Hawaiian immersion school right here in Hilo. And they come out just about every week to collect data in our site. So they actually collected data on phenology for us, looking at that fruiting and flowering, and did their own data interpretation. And they looked at how many introduced versus native species are fruiting and flowering throughout the year. So that was really awesome for them to get that like hands-on experience and also create their own data and their own analysis of that data. And they got to do a symposium at the end of the year. And so moving forward with that, we're continuing that collaboration this year, but we're also doing lots of collaborations with other local schools like Hilo High School. We just had a group out from Pahoa High School yesterday. So we're just trying to get people engaged with this space, talk about hybrid forest restoration. And honestly, my goal is for the kids to leave knowing 2 more plants. If they know that much, I'm really stoked. Because oftentimes they come and they don't know any plants. And of course, it depends on the school, their background, what class they're coming from. But oftentimes they'll come to the site and they don't even know ohia, which is one of the most common plants here. So just to get them engaged with the environment and learning about plants and starting to care about the plants that are around them. And if they can identify one more plant in their backyard and know its name and start to care about it in that way, that's fantastic.
We've also in the past and currently we've worked with the community college. So Hawaiʻi Community College, that's actually how I originally got involved. They bring out students and they collect data. A lot of times it's like biology classes or environmental science classes, or like the clubs on campus. So like one of the clubs that comes out is tropical forest ecosystem and agroforestry management. They'll come out and they'll like practice how to take data so they can apply what they're learning to the classroom with like real world stuff. We have other people from Hilo come out when they're interested, and sometimes they'll do directive studies and sort of make like their little own project, sort of offshoot of what we're doing. If they have like any burning research questions on their mind. So we've really have been trying recently to make the forest available to everybody. Like, you know, as long as you can safely do things in the forest, we want to be helping out. So we've been doing a lot of like different stuff, even like taking for classes that can't come out. We've been sort of taking the forest to them with different workshops and bringing out the greenhouse plants to them and teaching them about those. So we're really trying to do it as round as we can, because I had a kumu at Manoa who told us that no matter where we collect our data, the data belongs to that space. And it's our responsibility as scientists to be able to put what we're doing in the data with that and communicating that to the public.
That's amazing. I'm very excited to hear about all of the engagement with the community that you have. And in the chat, Rob also agreed and said that this sounds like a great way to get kids involved with the planet and to learn scientific methods too. Thank you so much. Thank you so much, Rob, for that comment. You know, the seeing coding used as a part of this process was actually surprising to me. And so I wonder, when you have groups that come to visit, do you go through some of the coding techniques that you use, at least on a very basic level, so that they know that that's an important part of getting into this field?
Yeah, that's something that we did with the Ka'umeke kids. We sort of helped guide them through their own data analysis. So they weren't coding in R because it's a little too complicated for middle school students, but they got to do their own analysis using just Google spreadsheets, and we sort of walked them through how they can filter their data and take these counts themselves. So they kind of had to do it the long way. They didn't get the shortcut of R, but I think that helps them better understand what exactly they're doing with their data, how exactly they're interpreting it when they have to manually count those yeses and nos themselves.
Absolutely. So where can we find your research? So you send your the grounded leaf particles after it's in the lab, it goes off for this analysis. When you receive that data back, what's the next step in that?
Yeah, so there's been a number of published studies that have come out of this project. Our most recent big report was in 2016, but hopefully we'll get another one of those out in the near future. And personally, I'm just working on a newsletter to start sending out to folks so that we can update people on the project, how the trees are doing, different outreach things that we're working on. So that's another just more informal way that we're looking to communicate our science, but we have a number of published papers on our website. So if you Google Likonapilina, will pop up, and we have a whole page dedicated to publications, so you can read more about the project there.
Wonderful. Well, I am very excited that you were able to allow us and me to actually be in your lab today to physically shadow you and share the amazing research that you do with the community and with the world. Now, next month, I will be with you in a dedicated session with a local school group. Can you tell us a little bit about that in our last minute?
Yeah, absolutely. So that's the Lanakila Learning Center. It's a alternative learning center affiliated with Hilo High School. So it's a group of kids that traditional learning structure just doesn't work super well for. A lot of them are much more hands-on learners. And so we've been really enjoying working with them. We actually have them coming out in the field this afternoon too, because they just really need to interact with their learning material. Just sitting and reading a paper or listening to a presentation doesn't work as well for them. So we're really excited to have them out in the field and come into the lab. And so for our session with them in the lab, which is next month, I believe, we're going to be focusing on our nematodes, which are this biocontrol that we're doing against Queensland longhorn beetle out in the field. We've been getting our trees infested with this burrowing beetle that cores into our trees and basically eats them from the inside out, but they actually don't eat it. They just destroy it for fun, I guess. Well, not for fun, but to rear their young. But we've been really getting decimated by these beetles. So we're working on rearing these nematodes that are found right here in Hilo in the soil. And they basically target these larvae and use this bacteria that they have a symbiotic relationship with to kill the larva, and then the bacteria creates the perfect nutritive environment for the nematodes to rear their young. And so then they can reproduce within this cadaver and go off and find another larva to reproduce within. So it's been proven pretty effective against the QLB, at least under the right conditions. We are very infested, so we're really needing to stay on top of it, but that's something that we're going to highlight with these kids. And they got to see some of our infected trees, and we're hoping to also get them to help us treat them with the nematodes eventually.
What an amazing way to learn and to contribute to this ongoing research project. Emma, Sophia, Rebecca, thank you for allowing us into your lab and to allow our virtual participants to be here as well. This is the Liko Napolina team here at the University of Hawaii Hilo. We look forward to our next session with you next month. Although it's dedicated, of course, we will be posting not only this session, but next month's session on our website, Shadow the Scientist, on our YouTube channel as well. And in our follow-up survey, I will share links to your most recent papers with everyone who was in this session today, and any other links that you'd like to help us spread the word also about the amazing works that you're doing. I'll be sure to include those. For now, we are so very grateful for your time and expertise, and we look forward to our next session with the Liko Na Palina team. Mahalo nui loa. Aloha.
Mahalo. Ahui ho.